Hi all, Dr. Clark here again, and today we're going to continue talking about cellular respiration. So remember last lecture I was talking about glycolysis and how sugars, carbohydrates, can be broken down without oxygen through glycolysis to obtain a little bit of ATP. I'm going to continue talking about that real quick because I mentioned the fact that animals produce lactic acid, and plants and fungi and some bacteria and protozoans produce ethanol or alcohol. Okay, I'm going to show you a little bit more on that. I think some of you are just like, well, how is it done? And so I just want to go through that in a little bit, in a little greater detail in just a second. But first, I just kind of want to introduce what we're going to talk about today. We're going to talk about pyruvate dehydrogenase or pyruvate processing. So remember, after glycolysis, after you break a sugar molecule, glucose molecule down, you break it into two 3-carbon pyruvate molecules. Well, then we're going to process that pyruvate further, and we do that through what we call dehydrogenase. And then after that, we're going to process the product of the pyruvate processing further, and that product is called acetyl-CoA, and that goes into the Krebs cycle. And this is a picture of the Krebs cycle. So I don't know if you can read this, but this top word says acetyl-CoA. Right? And so that is going to enter the Krebs cycle. The Krebs cycle is sometimes called the citric acid cycle also. And it's going to go through a bunch of different shifts. It goes through a bunch of different proteins. This is one giant biochemical pathway, and different things occur, like dehydration occurs, oxidation occurs, substrate level phosphorylation occurs, so ATP is going to be formed in one of these steps. You get these electron carriers throughout the steps, so NADH is formed, FADH2 is formed, and these are going to be really important for the production of ATP in the electron transport chain. But we'll go through this. I'm not gonna have you memorize all the steps and all the intermediates, but I want you to know the general process by which acetyl-CoA gets converted into energy-carrying molecules, NADH and FADH2, and then some of that energy is converted into ATP. Okay? All right, so before we get to the Krebs cycle and dehydrogenase, first I want to make sure that you understand that organisms can metabolize food without oxygen, and they do this often. Okay? So when there's no oxygen, the organism has to use glycolysis to produce ATP. So some organisms that live in anoxic environments, this is the only way they produce ATP. Okay? So they don't have an option to do cellular respiration and have oxygen in. Now, some of the products, some of the conversion for glycolysis without oxygen, one is called fermentation, and many of you eat or have drank the goods from fermentation process, but that's really breaking down sugar without oxygen being present. And as a process of it, you're still going to generate an electron carrier. By generating that electron carrier, you still have this energy bound up, and it can go to a new region of the cell, or a new portion of the cytoplasm, depending on the organism, and that electron can be used to form ATP. Okay? And we'll get to that point in a little bit. Okay? So, like all good cycles and like good biological systems, what is being converted will be converted back to its original material. So in this case, NADH is going to be formed from the processing of the glucose molecule, and then that hydrogen and electron will be donated to an electron transport system, and then the NAD is formed again. okay, or recycled. And for things like bacteria and archaea that are going to be doing this in their cytoplasm, because they don't have mitochondria, they don't have organelles. So this can still occur in the cytoplasm or across their plasma membrane, from inside the cell to outside the cell. This process can still occur. And we'll come back to this, and I'll show you some videos in a later lecture on the ability for organisms to do that. Okay, but first, when we're talking about bacteria, because bacteria do not have organelles, they've evolved well, depending on the species of bacteria, but they've evolved lots of different ways to go through fermentation, to go through the breaking down of glucose and then utilizing the energy of that to form ATP and the byproducts to make ethanol or alcohol. Okay. Eukaryotic cells, there's just a few types. Okay, we're going to talk about two main types, and that is the production of ethanol. Okay. And so yeast, which is a fungi, and plants do this, and most of you know this because we make ethanol out of corn and stuff like that. Some prokaryotes can do this, but that's taking pyruvate and converting it to acetaldehyde, okay, and then using acetaldehyde and converting that into ethanol. And so it accepts the hydrogen, okay, to form NADH, and you have ethanol that's left. Okay. And then, like I said before, that NADH can be utilized in the electron transport chain. Now, some bacteria have the capability of doing just this. Okay? They still have the capability of going through fermentation in the same way that yeast do. But they also have other ways in which they can do it. So I shouldn't say that the product of the bacteria fermentation is always ethanol. There are some other intermediate products that bacteria can form. In the case of animals, that pyruvate accepts that hydrogen atom. So the pyruvate gives up a hydrogen atom to NAD to form NADH. I'm sorry. Okay. The NADH that was formed through glycolysis, through the breaking down of glucose, you remember that electron carrier, it donates an electron and hydrogen to pyruvate instead of having an intermediate. So in the case of yeast, you have an intermediate acetaldehyde. Okay. Instead of the acetaldehyde picking up that hydrogen to form ethanol, in this case, pyruvate just picks up that hydrogen to form lactic acid. Okay. And so I'll show you how this is done in this diagram. Okay. So again, we already talked about glycolysis. We talked about converting glucose, breaking that 6-carbon molecule down into 2 3-carbon pyruvates, and the generation of ATP occurs from that. Now you have these 2 3-carbon pyruvates. In the case of fermentation by yeast and plants and things like that, carbon dioxide is going to be released, and acetaldehyde will be formed, and then the acetaldehyde will pick up the hydrogen and electron from the NADH that was formed during glycolysis to form ethanol. Similar process in animals. Same thing, glucose, 2 pyruvate molecules, NADH is donating an electron and hydrogen to form lactic acid. Okay? So I just want to make sure that you understand that this can still occur without oxygen, and you still have the capabilities of having some electrons flowing through the system. And in some cases you have intermediate molecules that can last in the system. Most of us have felt lactic acid or the result of lactic acid. If you go and just like do a sprint, run a 40, whatever, run a 100 meter or 100-yard dash, 100-meter dash, and your legs start burning, okay, that's the formation of lactic acid in your muscles. because you haven't supplied that muscle with enough oxygen for pyruvate to be processed through cellular respiration. So instead, some of that pyruvate is being processed and forming lactic acid. To form ethanol, most of us know how to do that. You just take a little glucose, add something like yeast and water, and stop any oxygen from coming in. So plug it up so there's no oxygen coming in. If you've ever made bread or beer or wine or anything like that, you would know that CO2 is produced, right? And you're going to eventually get alcohol depending on when the process is stopped. If you're attempting to get alcohol, if you leave it long enough, glucose, the sugar, will be converted into ethanol with carbon dioxide as a waste product. All right, so long way to get to where we're going. Okay? Now again, after glycolysis, there are pyruvate molecules. Now these pyruvate molecules still have a lot of energy. Remember, there are two of them, and they're both 3 carbon molecules. So there's still quite a few bonds in there, and we can, break those bonds and release the energy from them. And that is what cellular respiration does. So that pyruvate molecule needs to be moved into the mitochondria. So this is only occurring in organisms that have mitochondria, which is pretty much everything except for archaea and bacteria, and then some organisms don't have these capabilities. Some cells don't have these capabilities, but for the most part, all eukaryotes have the possibility of having mitochondria. Now some might not, but they have the possibility because they can have membrane-bound organelles. Now, so that pyruvate is moved into the mitochondria. Okay? It gets oxidized, it gets processed. So remember, oxidized means we're taking electrons from it, borrowing a hydrogen from it, and when you do that, you form acetyl-CoA. Okay? The enzyme that has this processing function is called pyruvate dehydrogenase, and so it's going to cleave off a carbon from each one of those 3 carbon molecules. So remember, you have 2 pyruvates from 1 molecule of glucose, so this is going to happen twice, and it's going to cleave off one of the carbons, and that carbon is going to bind with oxygen. That's where the oxygen comes in. That's where we, when we're taking in oxygen, breathing in oxygen, that oxygen is utilized to bind up to these free carbons that are coming out of pyruvate dehydrogenase and also the Krebs cycle. So the carbon leaves as part of a carbon dioxide molecule, and then you expel it. And the hydrogen and electrons are donated to an NAD molecule to form NADH. And then you have a remaining 2 carbon molecule, which is going to be bound to a coenzyme A, or what we call CoA. And so that's just kind of a carrier molecule, and it allows for it to move into the Krebs cycle. And the final compound, when you combine that 2 carbon fragment with coenzyme A, is acetyl-CoA, or sometimes people just call it acetyl-CoA, but most people call it CoA. Okay? And so this is kind of that process. We already talked about glycolysis, formation of pyruvate. Pyruvate gets processed, carbon dioxide leaves, and then you form acetyl-CoA. Now this is the interesting piece, and we'll talk more about this in a later lecture, but this is the part where pretty much all the other food components, biomolecules that you're going to consume, this is where they come in. So this process up here, glycolysis and whatnot, pyruvate processing, is really only for carbohydrates. If you're a protein, you're going to be broken down into amino acids. Those amino acids will be converted into acetyl-CoA. If you're a lipid, you're going to be broken down, your fatty acids chains are going to be broken down, and you're going to form acetyl-CoA. And so here's the kind of fork in the road. And if you have too much acetyl-CoA in your system, so you're not expending all of your energy. So you have a lot of ATP that's already being created, but you're not utilizing it, and so you got a bunch of acetyl-CoA kind of backing up because you've ate a huge meal or whatnot, and it's being converted into acetyl-CoA, and you're not going to burn it off. Acetyl-CoA is converted into fat. And this is why I tell you, it doesn't matter if you're going to eat a ton of carbohydrates or no carbohydrates. If you're eating more protein or more lipids than you can burn, it makes no difference where you're putting in the energy because it all comes to the same point, acetyl-CoA. If you have too much of it, it'll be converted to fat. If you don't have enough, it's converted to ATP and you'll start utilizing that energy. Okay. So here's how that intermediate electron carrier works. We kind of talked about this before. Remember when we were talking about photosynthesis that the electron carrier in photosynthesis is NADPH. In this case, it's NADH, and it's formed just like the other biomolecule processes or the enzyme processes that we talked about before, where you have a substrate that comes in and it's holding a hydrogen and electron. That causes the protein to change its shape, pushes the two molecules together. The hydrogen is transferred to NAD to form NADH. Along with that, electron goes over, the product is formed. Now this NADH molecule then can carry those electrons to another part of the cell, or in this case, another part of the mitochondria, which allows for that energy to be utilized later.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron, a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
Okay, so we've seen that many times when we were talking about enzymes. The process is the same. You get a shape change in the enzyme. You get one material passed to the next. In this case, it's just passing a hydrogen and an electron over from that original substrate to form a product. So it's a little bit different, but same premise in how these enzymes work. Okay, so let's look at the fate of acetyl-CoA. So we talked about pyruvate processing. So pyruvate is going to be broken down into acetyl-CoA. You're going to have two acetyl-CoA molecules. Carbon dioxide leaves, NADH is formed. Okay? Now the fate of acetyl-CoA really depends on how much ATP is in the system. Okay? If there's insufficient ATP, then acetyl-CoA will move into the Krebs cycle and start being converted into other electron carrier molecules and some more ATP. If there's plenty ATP in the system, then acetyl-CoA will be diverted or converted into fat. Okay? And it'll go through the fat synthesis where the carbon molecules are going to be bound in hydrocarbon chain to form a fat molecule. And so again, like I said before, if all food items that you're going to consume are going to come in as acetyl-CoA, then it really makes no difference if you're eating carbohydrates, proteins, fats, they're all coming in the same way. Now, to convert them to acetyl-CoA, that can take a little more energy, that can take a little more steps, it can take put a little more stress on things like your kidney, your liver, these kind of things, which we'll talk about in a later lecture. Okay, so let's look at that second step of oxidative respiration. So cellular respiration is the whole thing, but oxidative is meaning that oxygen is involved. In this case, oxygen is going to be involved by binding up the carbons that are being removed from these molecules to form carbon dioxide. Later we'll talk about how oxygen is also the electron acceptor, so it's going to pick up that electron and form metabolic water, but it's going to take a little bit before we get there. So the Krebs cycle, there is 9 reactions, and we're not going to memorize all of them, but since it's a cycle, you already know that the beginning molecule has to be returned at the end. It's just like the Calvin cycle. The beginning molecule has to return at the end. Okay? And again, like I said before, the Krebs cycle is sometimes called the citric acid cycle. Okay? The two are the same. So the first stage, take the acetyl-CoA molecule and bind it to a 4-carbon molecule to form a 6-carbon molecule. Okay? Then, taking that 6-carbon molecule, you're going to remove carbon molecules to form CO2. And as you break the bonds, you're going to have electrons that are going to be donated along with a proton to NAD to form NADH. And also, there's enough energy from the breaking and the rearrangement of those carbon chains to form ATP, to phosphorylate ADP. And then at the end, you're going to return the original 4-carbon molecule. And in doing that, you again have to rearrange some carbons, okay? And you're going to break bonds and make it, more streamlined. And in doing so, electrons will leave, protons will leave, and they'll be picked up to form NADH and FADH2. FADH2 is just another type of electron carrier. So both of these are electron carriers, and they're going to be useful when we talk about the electron transport system. So here you can see the whole kind of system, exactly what was just written out for you. Acetyl-CoA is coming in. The CoA, or the coenzyme A, is going to be removed right in the first step, and the 2-carbon molecule, the acetyl portion, is going to be bound to that 4-carbon molecule. Then that 6-carbon molecule is going to go through alteration to the shape. Carbon's going to leave. You're going to form a 5-carbon molecule. More carbon's going to leave. 4-carbon molecule. And in the process, these electron carriers will be formed and ATP will be formed. That 4-carbon molecule is not the same 4-carbon molecule, so it has to be changed, and the shape has to be changed back to its original starting material. And in doing so, electrons are removed, hydrogen ions are removed, and you get NADH and FADH2.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Okay, so I want to make sure that you're understanding that this is not just hypothetically happening in solution in the mitochondria. Remember that each one of these steps involves at least one enzyme. Right, so remember, acetyl-CoA is going to be able to fit into the active site of a certain enzyme. That enzyme is going to change its shape. When it does so, it's going to kick off the CoA, the coenzyme A, and bind the carbon dioxide or the 2-carbon molecule to the 4-carbon molecule to form a 6-carbon molecule, and then on and on and on. So each one of these steps, there's at least one enzyme, if not more than that, to form these intermediate molecules until you return the starting material. Okay, so just make sure that you understand that this is an enzymatically driven process. Now, when this is all said and done, we've done glycolysis, produced pyruvate, pyruvates produced acetyl-CoA. Acetyl-CoA is going to be entirely consumed. Okay? There's nothing left of it. Those 2 carbon molecules are leaving as carbon dioxide. So when it's all said and done, it's all been consumed, and this is what the energy from that original glucose molecule is converted into. 4 ATP molecules, 10 NADH molecules, and 2 FADH2 molecules. Now, if you remember back from photosynthesis, how much energy did it take to make or to produce 1 molecule of glucose? I think, you know, when it was all done and said, I mean, I rounded it up to 60 ATP molecules, but it's probably around 54 is on average, 54 molecules of ATP to produce a single molecule of glucose. And so far, we get 4 from this process. And, oh, I'm sorry, I take that back. We get 2 from the Krebs cycle, and we get 2 from glycolysis to form 4 ATP molecules. So we're taking 54 molecules of ATP and we're getting 4 out. So it's not the ATP that's coming from glycolysis, pyruvate processing, Krebs cycle. That's not the important pieces. The important pieces are here. The 10 molecules of NADH and the 2 molecules of FADH2. These are the electron carriers. This is what's going to drive the production of ATP. These are the key components of all of the material that we went through, all the proteins, the enzymes, and the intermediates that we just went through the last 2 lectures. The purpose of those is to create these electron carriers, so then these electron carriers can be moved into the electron transport system and generate real ATP. Okay. All right, so we'll come back and look at the electron transport system next time.